Welcome to AP Biology. Today's presentation is going to be on eukaryotic control of gene expression outside of the nucleus. So one of the places where DNA expression gets controlled is by small interfering RNAs, or also known as microRNAs. These are short segments of RNA that have the complementary sequence to messenger RNAs. So they will bind to the messenger RNA and prevent it from being read by the ribosome. And so they prevent basically translation from happening by binding to these messenger RNAs. They allow messenger RNAs that are needed to be expressed, but they stop messenger RNAs that are not needed from being expressed. And so basically they trigger the messenger RNA to not be read. The exonucleases are eaten from the ends, and if you're not read quickly, then you're destroyed. And so they pretty much silence genes by preventing them from being read quick enough. Another way of stopping translation is you can bind, proteins can bind to the messenger RNA and prevent the ribosomes from being able to grab onto the messenger RNA. So they prevent the ribosomal subunits from actually grabbing on and scanning and looking for the AUG start codon. And if you prevent those subunits from reading, once again, endonucleases are going to destroy it before it can actually get read. So this prevention process doesn't have to last very long. If it prevents it from being read for a minute, too much damage is going to be done and the messenger RNA will not be functional. The other place where we can control gene or protein, the amount of protein in the cell, is we can, after it's translated, proteins often have to be modified, so they have to be folded or cleaved. Portions of them have to be cleaved off, or maybe you're going to add a sugar or a fat group to them. Well, if you prevent any of those processes from happening by blocking the sugar, for example, from getting to, or the enzyme from getting to the Golgi, you're going to stop that protein from becoming a functional enzyme. And finally, there's two things. There's proteosomes, which break down proteins, and ubiquitin tags, which identify proteins to be broken down by proteosomes. Those are like death tags. So like if a protein is not needed, if there's an excessive amount of a particular protein in a cell, ubiquitin will tag it for destruction. And if it gets tagged by ubiquitin, proteosomes will come along and break it down and chop it up into tiny little pieces. Those degraded proteins can be broken back down into the amino acids and then be reused to make other proteins. So there's lots of different ways that we can control gene expression, from transcription to post-transcription to prior to translation to post-translational. So gene expression is a little more complicated in eukaryotes than it is in prokaryotes.